Good morning, good afternoon to those joining us from the United Kingdom and Mexico and or elsewhere. My name is Antonio del Rio. I am the director of the Center for Mexican Studies, UNAM in United Kingdom, and we are pleased to and we are pleased to host this seminar from the students organized with the Mexican Society of Students here in United Kingdom is MexSocUK. And I am happy to introduce to Farid Bustos, is the president of this MexSoc. The floor is yours, Farid.
Hi, thank you, Dr. Del Rio, for this introduction. And as Dr. Del Rio said, we're delighted to host this session as part of our ongoing efforts to support academic exchange and highlight the work of Mexican researchers in the UK, so we can foster dialogue and innovation and sustainability. Today's seminar is entitled Developing Sustainable Superhydrophobic Coatings from Pine and Palm Pollen. And the speaker is Brenda Resendiz Diaz. She studied textile engineering at the National Polytechnic Institute in Mexico. She developed a strong foundation in textile manufacturing and material science. She has then gained some industry experience in textile dyeing, productions, and materials development. She is currently a PhD student or researcher at the School of Engineering and Material Science at Queen Mary University of London, and she's a member of the Creek Research Group. Her work focuses on development of sustainable superhydrophobic materials, particularly food-grade water-repellent coatings for environmentally responsible packaging applications. In addition to her academic research, she's a co-founder of EcoBarrier Limited, a startup dedicated to sustainable superhydrophobic coating technologies. Her work bridges scientific research and innovation, contributing to the development of greener material solutions. Brenda, thank you very much for joining us today, and the floor is yours.
Thank you, Farid, for the introduction. And also, it is a pleasure to be here and share a little bit about my knowledge and some of my work now here in Queen Mary. So I'm gonna start sharing my screen. So I think now is everyone can see my presentation. So yeah, the title for today's presentation is Developing Sustainable Plant-Based Highly Water-Repellent Coatings Using Hydrophobic Pine and Palm Pollen. My supervisor here in Queen Mary is Dr. Colin Crick. And yeah, let's start. So my presentation is divided in 4 sections. First, we're going through the introduction, where I'm going to explain you about the problem that we are facing and how we are going to tackle this problem, some fundamentals of superhydrophobicity, and then why I choose, in this case, pollen for the fabrication of the coatings. Then we are going through the experimental section, where I'm going to explain you about the materials and methods, then the results and discussion, and finally the conclusions and future work of this research. So the challenge, as we know, plastic waste are becoming an important issue to tackle, as they are, some of them are difficult to recycle and they can persist in the environment for at least 400 years. And around 85% of plastic packaging worldwide ends up in the landfills. And some of the paper products that can substitute plastic, they are not water resistant, so they need a non-recyclable lining layer, which makes them more difficult to recycle or even biodegrade in the environment. Also, there are other challenges like non-biodegradable or toxic materials like greasy-proof or PFAS, which are the materials that are fluorocarbon-based, and they are mainly used in some food packaging containers, especially like in fast food packaging containers. So yeah, they want to ban these products by using other alternatives. Another example can be polyvinyl chloride, which is also used in bottled oils and films. So I just want to show how we can make a superhydrophobic surface, or what are the fundamentals. So let's imagine we have a surface, and then the surface is coated with a repellent material or very hydrophobic material. And we need to increase the roughness to create this superhydrophobicity. So we need to combine two important elements, which is a hydrophobic material and roughness. So a good example of this, we can find it in nature. For example, the lotus leaves, where they have these it's like micro structures or very rough structures that are covered with wax microcrystals. So this helps to repel water. So researchers want to mimic these features by using synthetic materials. So here is an example of an image where they use small pillars, and they are covering with a repellent material to create this high water repellent surface. So in my project, the potential solution to this is to use only 100% plant-based materials, where we use a roughening agent, which is going to be the pollen, and it's going to cover by a wax coating or paper. So these coatings can be biodegradable and can have similar properties to plastic. So, but why I choose pollen? So actually pollens are the seed of the genetic material of seed plants, and they also provide nutritional values, and also they have eco-friendly benefits. Sporopylenexin, which is a part of the pollen, which I'm gonna discuss later, has been explored as a microcarrier for oral drugs and vaccines. So actually pollen, you can eat it and it's not harmful. And also it has the potential of tunable wettability, which means that we can actually play around with the chemical groups of the pollen, turn it from hydrophobic to hydrophilic and vice versa. And also they have a very rough microstructure, so we can also use that for our coatings. So here is an example of the pollen structure. So actually pollens, they have a pollen coat, which is made of lipids and waxes. Then we have the exine part, which is very strong, two different chemical treatments. And inside we have the intine layer and the genetic material. So in the genetic material, it's full of proteins, and proteins are very hydrophilic. So for us to try to improve hydrophobicity, we need to remove that proteins for the pollen. So we use an acid treatment to try to remove all that proteins, so we can keep just the pollen coat, which is hydrophobic, and the exine layer, which is gonna give you the rough structure. So for the material selections, how we can achieve this system. So I was inspired on the previous work of my research group, where one of my colleagues, Dr. Emma Sadler, she used Lycopodium clavatum spores. So I was looking for other pollens and other methodology where I can modify it to turn it hydrophobic. So I modified pine and palm pollen species. Then for the low surface energy material, I used two different types of waxes, plant-based waxes, Carnauba and soybean wax. And for a solvent, lower environmental impact. So in this case, I use ethanol. So for the pollen treatment, what I do is use phosphoric acid to remove all that proteins that I mentioned before at 75 degrees for 20 hours in refluxing. And then I use washing and filtration with DI water or distilled water to remove all the excess of acid in my samples. And then I dry it at 75 degrees overnight. And then you can observe here is the hydrophobic pollen. From the picture, you can see that there is a yellow pollen and it starts absorbing water immediately when I was placing a water droplet. But then after this treatment, that we remove all these proteins, it starts to turn more hydrophobic. So for the superhydrophobic fabrication, what I do first is take that sample and just use a pestle mortar to try to make a fine powder. And then I mix wax and ethanol to make a proper dispersion. And then I mix pine or palm pollen on ethanol. And then I just sonicate it for 15 minutes to have a more stable dispersion. I use spray coating as my main technique, and I anneal the wax to cover all the features of the pollen, and then we obtain a superhydrophobic coating. So for the characterization of my coatings, first I do some scanning electron microscope, so it will help me to see the morphology of my pollen. Here we have how the original morphology of pine and palm pollen. So from the original pine pollen, they have a vesicate shape. So it has like two sacs and a main sac. And for palm pollen, it has like a boat shape, but also it has some porous structure. But when I received my samples, actually pine pollen was broken, so I couldn't see or define the structure, while palm pollen, it was easy to see because it was a complete, the complete morphology. And actually, if you can see here in figure F, there is some, like, I would say the proteins that are inside the pollen. So here it's unfolded pollen. And as I receive my samples, the size of pine pollen broken ranges for 7 microns to 4 microns. For palm pollen, it's around 20 microns. So actually, most of the pollen size rounds in general around 20 microns, but when it's broken, it could be a small size. But and after being treated, both pollens, I can see differences, especially in palm pollen. So there is an empty cavity, so that means that the treatment was working, while in pine pollen, it was kind of difficult to observe because it was already broken, so I couldn't see if actually the treatment was working, but that's why I need to confirm it with other characterization. So that's why I use FTIR, so Fourier transform infrared spectroscopy, to know if the chemical groups present in my samples. So what we want to achieve is actually reduce the OH groups or hydrophilic groups present on the sample. So we can observe these at 3,300. So they actually reduce from the untreated to the treated. So it was observed here. And we keep the two important peaks here, which are the CH stretching, which relates to the hydrophobic properties of the materials. So it was maintained at 2,900 and 2,800. Similarly, palm pollen has a similar behavior, where we maintain the CH stretching and we reduce the hydrophilic groups. Also, it was important to know the nitrogen percentage. The nitrogen percentage will give you an estimation of the protein content in the pollen structures. So here is the percentages of nitrogen, carbon, hydrogen. So from the untreated and treated pollens. So we can observe here that when it was untreated, we have 12%, and after the treatment, it was 2.6%. Same for palm pollen, it was 29.8%, and after the treated, we reduced it at 4%. So actually, the treatment was removing almost completely all the proteins here. So for giving this, I multiply the nitrogen for the factor 6.25. And here also in the carbon percentage, there was an increase here, and also for palm pollen. Also, it was very important to see the waxes chemical composition and also their crystal structure. So I did, again, FTIR, which confirms the CH stretching. So they are very hydrophobic. Actually, you can observe here that soybean wax and Carnauba wax has similar peaks. Actually, they are have very similar chemical structure. But when we go to the X-ray diffraction, which tells you about the crystal morphology, here Carnauba wax have two diffraction peaks at 21 and 24, or 4.2 Armstrong and 3.8 Armstrong, which corresponds to the orthophobic phase. This orthophobic phase relates to a specific crystal structure where it has all the axes are different length, but they are perpendicular. So this is very common in waxes. But for soybean wax, we have similar peaks at 21 and 24, also at 19, but there is one peak before 10 degrees, which is related to the presence of fatty acids, which reduce the crystallinity in our material. So actually, soybean wax is more like a semi-crystalline or amorphous wax, comparing to Carnauba wax in this case. I also perform DSC and TGA. So DSC and TGA will tell us the thermal properties of our materials. So at which point they start to melt and at which point they start to crystallize. So for both waxes, they have one melting point at 81 and soybean wax at 52. So actually, because of the crystal structure of soybean wax, the melting point is expected to be lower, and the crystallization of Carnauba wax is 78, for soybean wax is 34, and for TGA, the decomposition is around 250, and for soybean 300, and they start losing all its weight up to above 400 degrees. So yes, Carnauba wax has a higher crystallinity, which means that we will have a higher melting point, and soybean wax is getting more amorphous, so we lower our melting point. So for determining a superhydrophobic surface, we need to measure the water contact angle and sliding angle. So for being superhydrophobic, our water contact angle should be above 150 degrees. And when we have a sliding angle above 90 degrees, that means that our surface is getting more sticky. So for getting a superhydrophobic surface, the sliding angles mostly should be lower than 20 degrees, 10 degrees. So here is how we calculate the sliding angle. So actually, it's the position of how we tilt the surface. So I measure the water contact angle of a pine pollen Carnauba wax combination, palm pollen Carnauba wax combinations. So as we can observe, when I use 80% to 40% weight percentage of pine pollen, we have contact angles above 150. And when I increase the amount of wax, then I'm getting a more flat surface. So it's more sticky and the water droplets don't roll easily. In the case of palm pollen, actually was more like in the middle, so between 70 and 40% weight percentage, I have a contact angle above 150 and sliding angles between 20 to 17. And when I increase the amount of wax, then again, I'm reducing the contact angle because I'm getting a more flat surface, like all the wax is covering more features of the pollen. So here is when I use soybean wax. I want to observe also how, depending on the wax that I'm using, also it will change the properties of my coatings. So for pine pollen soybean wax combinations, actually I can observe that it was from 80 to 50 when I achieved 150, but the sliding angles were a little bit higher. So which means that maybe the crystallinity of the wax could also affect, but maybe might be also the morphology of palm pollen that could influence on the contact angle. So when I use a palm pollen, also I can observe that the water contact angle was lower. Even I couldn't achieve superhydrophobicity. Most of them were very sticky samples. So I can see that maybe this system between palm pollen and soybean wax was not the best. So I did water contact angle over time. So this will tell me how is superhydrophobicity maintained. I just place a water droplet on the top of the surface and I leave it until 15 minutes. I leave it until 15 minutes because the water droplet starts to evaporate. So it could be just arranged. And then I can observe that actually pine pollen with Carnauba wax was more stable. So it was superhydrophobicity. It was superhydrophobic for almost 15 minutes, while the pollen samples start to decrease the contact angle, especially when I was using soybean wax. So when I was using soybean wax, I can see a decrease on the contact angle almost immediately in some samples. And also we can observe here an SEM of my coatings. So when I was using 50 weight percentage of Carnauba wax, and when I was using too much wax, you can observe here how the wax cover all the areas of all the rough structures of the pollens. So yeah, here if we can observe how is everything covered. Here is a video of how my coatings looks like. They actually brown in color, which sometimes could be a challenge because in some food packaging, which is gonna be one of the main applications, they want it more transparent or white in color, but we were thinking that maybe paper, it's also brown, so we can also like find this combination. But yeah, this is I do it in glass slides for testing. But yeah, we can see here a 50 weight percentage pine pollen Carnauba wax coating, and here is 50 weight percentage pine pollen Carnauba wax coating, which it was the ones with higher water contact angles. So here's what I'm saying about the rolling, that you can observe that actually the droplets roll very easily through the surface. So they have this self-cleaning action and also anti-fouling properties. So for my conclusions and future work, so we first successfully demonstrate superhydrophobic coatings using these two specific pollen species, so pine and pine pollen. And we can obtain water contact angles above 150 degrees and sliding angles less than 13 degrees. And long-term superhydrophobicity also will depend on the system we are using. So we need to know the characteristics of our materials, especially of the waxes, and also it will be dependent on the pollen species. And novel applications of pollen microstructures and can be applied for a superhydrophobic coating. So hopefully in the future we can see more sustainable materials and maybe the use of pollen in other applications like coatings. For the future work, I'm currently studying the impact of the broken structure. As I mentioned before, one of my samples was broken. So I want to see if actually broken will benefit to at the long-term stability of my coatings. And also I am looking into the resilience of the coating. At the moment, they are very powdery because they are on glass. So I also want to see and perform some experiments in paper and cardboard so we can see how it's gonna be applied for a food packaging container, for example. So I just wanna mention also a little bit about eco-barrier. So eco-barrier, we spin out as a company last March, so no, early this year, sorry. And we have 2 patents for the technology, so that are filed in March 2024. So we want to make more sustainable coatings using 100% plant-based materials. And we have this advanced formulation using plant spores and pollen. So these are my references. And also I would like to thank my research group, CRC group, because they are always supporting me and helping me during my project. Also to Queen Mary University of London and Fundación Politécnico for funding. Also, if you would like to connect, I share my LinkedIn and my email. And thank you so much for your attention. And if you have any questions, please feel free to ask.
Thank you very much, Brenda, for such an interesting and insightful presentation.
Thank you.
As Brenda said, we will now be open for questions. If you're joining us via YouTube, please feel free to post any questions you may have in the chat, and then we'll do our best to address as many as possible. Professor Del Rio, do you have any questions you may address to Brenda, or should I?
Farida, yes, I have some questions. One is about the role of the initial conditions of velocity and volume when the drops start to fall. You
Oh yeah.
present very nice images, but the tip is not static. Then the initial condition and volume condition are different.
Uh, well, this video it was just with the purpose of showing the water repellency of the coatings, not actually a proper measurement. But yeah, I have all the conditions, but for the sliding angles that I can share with you if you would like. Yeah, this work is gonna be published hopefully this year. So yeah, I'm also happy to share all the results.
Perfect. Another question is the sliding angle in the PELM case was not monotonous. You have an increase in this behavior. Have you an explanation of that?
Do you mean like I'm gonna share again my screen so we can see. Do you mean from the graphs here?
Yes.
Okay. Yeah, so it was not very consistent as I can observe. So for example, we have 29, 24, between 40 and 50, but then it starts between 90 when I was increasing the amount of wax. But here at 80, it was more of the pollen. So probably it's because not all the wax was covering properly all the pollen, so that maybe cause like the water droplets to stick in some sections of the coating. So probably there could be some inconsistencies as I am using a spray coating. So sometimes spray coating could be like not very uniform coating, or maybe there are some spaces where it could be not properly covered by wax, for example.
Okay. Thank you. There are the two questions that I have, Farid. Thank you, Brenda. Very nice presentations.
Thank you.
Thank you, Professor. I've got a couple of questions myself. Brenda, the first one would be, I said there's a difference between your two wax samples. One's a little bit of a crystalline sample, and the other one, as you said, it's amorphous. I
Yes.
can see that there is some relationship between these two. That the Carnauba wax, they show a little bit more of a ideal water contact angle and the sliding angle. Do you think this has to be solely because of the crystallinity, or it's also a matter of coupling the wax with the pollen sample?
I mean, I think could be both, but also I think the crystallinity, it's also something that I think we need to consider, because yeah, like the crystallinity depends also on the structure of the of the of the waxes. So some of the waxes, they also have some additives on it. So it could be also that. But yeah, I don't know if I answered your question or.
No, yeah, you did answer my question. And the other one would be, you mentioned that and you showed us actually that the the materials are brown in color.
Yeah, true.
For this, as you said, the food industry is quite strict on their colorations. They either want it transparent, clear, or white. Do you think that if you change coloration, the most common thing to use it's pigment? You want white, it would be titanium or dioxide. Do you think this would change properties of the wax? Is there any other way
Yeah.
to overcome that?
Yeah, probably. I think it will depend on the type of pigment. So if the pigment is hydrophobic or is hydrophilic, we were thinking also like some kind of bleaching, because yeah, they are very brown. But yeah, it will change also the properties of the material. Like we will need to test how this will affect in the pollen structure. But yeah, I mean, we are trying to make more natural things, and I think the way to do it is to try to use the materials as they come from nature. So yeah, I think there is a way where we can use these materials without doing more modifications, because also what they were saying in industry is that they don't want to they don't want to modify chemically the materials. They just want to use it as it is. So I think this is more a simple way, and also we are not trying to chemically modify, for example, the waxes, or it's just taken from as they are and then combining to a formulation and apply it.
Okay, thank you. Yeah, no, it makes a lot of sense just to use the waxes, the materials as they are.
Yeah.
It will slash cost a lot, and it would be more sustainable and green in some way.
Yeah, exactly.
And I saw, well, just a bit of a FTIR nerd or curious about it. I saw that also in your FTIR spectrum. I said that the double bond for well the carbonyl group at 1774 had a slight. It
Here.
basically disappeared. At it's the 1714 bond. It basically disappeared. You think that that also is helping to the hydrophobicity?
Yeah, like I didn't want to like specify more, 'cause sometimes maybe some of the community will not be very familiar with the technique, but yeah, just wanted to show, okay, we want to focus on this. But yeah, here in the other pics that I also have here, yeah, there is a presence of esters and carbohydrates, and they are removed after the treatment. So actually, if there is a remove of carbohydrates, then that means that actually we are also reducing the hydrophilic groups. But yeah, there are other peaks here that also contributes to, or it give us more information about what is happening in the structure of the pollen.
Yeah, thank you. I think those are my questions. That was a very interesting and, at least for me, very entertaining presentation. Thank you very much.
Thank you.
And I don't know if there are any questions in the YouTube chat.
No, we have no question for it.
Okay, I don't think there are any questions. Again, thank you very much, Brenda, for your very interesting slides, your presentation. It's very interesting to see that we're trying. There are a lot of efforts to reduce the carbon emissions and try to make everything more green. Congrats, it's very, very interesting, and thank you very much again.
Yeah, thanks for also the invitation to be here. I mean, quite happy to also share a little bit about my research. I think it's good to show to the community what we want to achieve, and yeah, like now in this area, because also I think it's very niche, but yeah, it's very, very interesting, and I'm happy to explain a little bit about it.
Well, thank you very much, Brenda. Thank you very much, Farid. And thank you, everyone who joined us. We hope to see you at our upcoming seminars organized in collaboration with MexSoc and the UNAM Center for Mexican Studies. Have a wonderful afternoon, and thank you for being part of this academic community.